Hello, this is Matt Dean with A+ College Ready, and today we're going to talk about the immune system. The immune response is the way in which your body recognizes and defends itself against bacteria, viruses, and other pathogens that are foreign and harmful. We're going to talk about two main divisions of the immune system. The first is the nonspecific, or also known as the innate immune response. It's the defense system with which you're born. It protects you against all antigens, all pathogens. It involves barriers that keep harmful materials from coming into your body. This is the first line of defense. The second division is the specific, also known as the acquired or adaptive immune response. Once an antigen has been recognized, this part of your immune response creates an army of immune cells designed to attack that specific antigen, that specific invader. This type of immunity also creates memory that makes future responses against a specific pathogen or antigen more efficient and usually prevents the infected individual from showing any symptoms upon further infection. Let's first talk about the nonspecific immune response, and let's talk about the components of that immune response. The most important part of the nonspecific or innate immune response is the skin. It's obviously a barrier, it's like a wall, but it also creates chemicals such as urea and sweat, which help to kill pathogens or at least to slow down their growth. Stomach acid is also part of the nonspecific immune response. The low pH in your stomach, somewhere around pH 2, kills most pathogens that enter into your body. Interferons are groups of signaling proteins. They're made by white blood cells, and they help your body fight infections caused by general viruses, bacteria, and other kinds of pathogens. In addition, you have natural killer cells, sometimes called NK cells. These are immune cells which fight viral infections and also tumor formations. They recognize stress cells in the absence of antibodies and a major histocompatibility complex, MHC. And this allows the immune system to work much faster than it normally would. There's also complement. Complement is a complex system of more than 30 different kinds of proteins that act together to help eliminate infectious microorganisms. They cause lysis or bursting of foreign and infected or infected cells, and then the phagocytosis or ingestion of those foreign particles and debris. Another part of the nonspecific immune response is inflammation, and this is where plasma and leukocytes or white blood cells move to a damaged area. It's often stimulated by the release of histamines by a type of cell called mast cells, mast, M-A-S-T, and these are cells that are located in the damaged area. Another part of the nonspecific immune response are your normal flora. Now, in most cases, the word flora refers to plants or flowers, but in this case, we're talking about bacteria, the normal bacteria that live in your body in places like the digestive tract and the reproductive tract of women. These bacteria live there, and there's so many of them and they take up so much space that they inhibit the growth of other things that might be pathogenic, like yeast, for example. Macrophages are yet another part of the nonspecific immune response. These are a type of white blood cell that engulf foreign invaders. Also, they engulf dead or damaged body cells and help to get rid of those by intracellular digestion. Your saliva and your tears are also part of your nonspecific immune response. Both contain an enzyme called lysozyme, which helps to break down the bacterial cell walls and get rid of those from your body. Next, we'll talk about the specific, also known as the acquired or adaptive immune response. And before we do, there's a couple of really important terms to be familiar with. The first is the term antigen. An antigen is a foreign substance, almost always a protein, which can stimulate the immune response to initiate a specific immune response. And the second one is an antibody. Antibodies are proteins made by cells in your own body called plasma cells, and they're made in response to a very specific antigen. The antibodies will bind to that antigen and help the body neutralize it and get rid of it in one way or another. Next, we'll talk about the two different divisions or parts of the specific or acquired immune response. First one of those is called antibody mediated, or also sometimes called the humoral immune response. And this is when immunity is conferred to an individual through the activity of cells called Bcells or B lymphocytes. These cells produce antibodies that circulate through your body, through your fluids. They're made in response to a specific foreign substance or antigen. They attach to it and help your body to rid itself of it. This process is known as humoral immunity because these antibodies are large proteins and they can't actually enter into cells like infected cells. They can only kill germs or pathogens when they're floating around in the body's fluids. So humors are fluids. The second part of your immune response, your specific immune response, is called the cell-mediated immune response. And this is the immune response in which active cells called cytotoxic T cells, destroy your own cells which are infected with some type of internal or intracellular pathogen, usually a virus. And this helps to keep those infected cells from spreading the germ to other cells in your body. This diagram is a really good one to understand, and we're going to come back to it in just a second. But it outlines how both parts of your specific immune response, the humoral antibody-mediated part and the cell-mediated part, how they're activated and also how they function. So let's talk first about antibody-mediated or humoral immunity and sort of how it works. First of all, it's activated when a macrophage or some other phagocyte, a cell that engulfs germs, engulfs those things, does what's called intracellular digestion on them, using lysosomes to break them down into pieces. And then it displays some antigens, some proteins from the pathogen on its own cell membrane, using a protein, and this should say protein here, not program, a protein called the MHC2 complex. And that process of taking the pathogenic antigens, pathogenic proteins, and displaying them on the macrophage's own cell membrane, that process is called antigen presentation. And it's a type of signaling. The macrophage here is signaling other components of the immune system to the presence of this particular pathogen, and it's telling them to become active and help to eliminate that pathogen from the body. So the macrophages will interact with a type of cell called a T helper cell. Some people call them helper T cells. These cells have binding sites for antigens, and only helper T cells with a binding site that will attach to this specific antigen will bind, and they'll become activated. The macrophages will also release a type of cell signal called interleukin-1, which helps to activate these particular helper T cells. Once they're activated, helper T cells stimulate specific B cells or B lymphocytes. B lymphocytes that can produce antibodies that are specific to this particular antigen, this particular pathogen. And again, they're going to release a signal called interleukin-2. Only B cells with receptors for this specific antigen are activated. And they're going to make antibodies that are very specific to this antigen. Once the proper B cell type is found and it becomes active, that particular B cell type is cloned. It makes lots of copies of itself. It goes through what's called monoclonal selection. Before we finish that up, here we can see the first part of this process. This is our macrophage, and it is engulfing, through phagocytosis, a pathogen, maybe a virus. It's going to break it down into pieces, which is what these little triangles represent. It's going to use this protein called the MHC2 protein to present those antigens on its own cell membrane right here. A helper T cell with a receptor called a CD4 receptor, a receptor that can attach to and bind to this specific antigen, will attach. And once it does, with the help of signals released by the macrophage called interleukin-1s, this helper T cell becomes active. I compare these helper T cells to Paul Revere. They're sending out the message not that the British are coming, but that the germs are already here. And here's what they look like. Go get them. So let's finish up the antibody-mediated or humoral immunity. Most of those cloned B lymphocytes or B cells mature, and they become a type of cell called plasma cells. These are essentially cells that are specialized to make proteins, in this case antibodies. Antibodies that are designed to fit to the particular antigen in question. once the infection is over with, you don't really need all these plasma cells anymore. So most of the plasma cells undergo a process of programmed cell death. That's called apoptosis. So they go away fairly quickly. Some hang around for a while, but most die pretty fast, and the antibody levels drop back down to almost zero. But some of those activated B cells, they never became plasma cells in the first place. They instead became a type of cell called a memory cell. In this case, a memory B cell, or a memory B lymphocyte. So if that antigen ever gets into your body again, a lot of the initial steps are skipped. The secondary immune response happens in a much faster way. It's also a much bigger response. And that secondary response happens so much faster and so much larger that typically you don't get symptoms upon the second, third, fourth exposures. These memory cells can stay in your body for maybe the rest of your life. So we might say that once you've been through that first infection and you're over it, that you're immune. You're immune largely because of the presence of these memory cells. So here we can see this happening. We can see the B cells. These B cells can either be stimulated by antigen presentation from a helper T cell, or in some cases the antigens will actually, freely floating antigens will bind to receptors on the B cell. Notice that the antigens only fit to specific receptors, and only the B cells with those antigens are bound and activated. Those cells are activated. They now go through monoclonal selection or clonal expansion. Most of those cells differentiate and become plasma cells. They begin to make antibodies, which eventually will attach to the germ, the antigen, and help your body to get rid of it. When the infection's over, they go through programmed cell death or apoptosis, and they're gone. Some of those activated B cells, though, became memory cells, and those are going to be with you probably for the rest of your life, and they're going to essentially help you to become immune to that specific antigen or that specific pathogen. Here we can see what an antibody looks like. Again, an antibody is a protein. You're sometimes going to hear antibodies called immunoglobulins. That's why you'll sometimes see them abbreviated with an IG. Notice that they have a binding site that looks very much like the active site of an enzyme. The antigen has to fit in there almost like a lock and a key. And that's why antibodies are so specific. An antibody for a particular antigen is not going to work on another antigen. They're very specific. And that's why you might get several versions of a cold during your lifetime. Those different colds are caused by different versions of the cold virus. The antibodies to one of those cold viruses won't work against other cold viruses. A couple of other things to know about antibody-mediated immunity. Antibodies, again, are proteins. They bind to pathogens, and they mark them for death. They can cause a couple things to happen. One of them is opsonization. And this is the process in which the antibody binds, and it sort of is like a signal to other parts of your immune system to come and kill this thing, come ingest it and eliminate it. It's a signal to phagocytes like the macrophage. Antibodies can also cause agglutination, and this is when a lot of the invading cells are bound to antibodies, and it causes them to clump together. And that just makes it easier and more efficient for the macrophages and other phagocytes to get them out of your body. Our bodies have a limited number of genes which code for antibodies. We don't have that many genes for antibodies. But even though we don't have very many antibody genes, the average human can make at least 10 to the 12th different kinds of antibodies. Again, antibodies are proteins. Proteins are coded for by genes. We only have a few antibody genes, but we can make millions and millions of different kinds of antibodies. The main way that happens, the main way it's possible, is by something called alternative DNA splicing. This occurs between transcription and translation. It's a process called RNA processing, and this is essentially when during gene expression, it results in a single gene being able to code for many different proteins. We have these coding parts of our DNA called exons and non-coding parts called introns. What's an exon in one situation may end up being an intron in another situation. When you guys get to our unit on genetics, you'll get much deeper into this. But for right now, just realize that one gene can code for different proteins. So even though we only have a few genes that code for antibody proteins, we can make millions and millions of specific different antibody proteins. Here we can see alternative processing happen. So here's a piece of mRNA that was coded for by a gene. Notice the exons are labeled with letters. The introns are the non-coding sequences. And notice that between transcription, which is what we're looking at here, and translation, which we're looking at here, we get this process called RNA splicing or RNA processing. And in this case, the introns are removed. So here we're left with a mature piece of mRNA, which will then be translated and used to make a specific protein with a very particular shape. This might be an antibody. Here, though, is the same gene. But in this case, there's something different going on in your body. So that mRNA is processed a slightly different way. Notice that a sequence of it is removed that wasn't removed on this side. So now we get a different mature mRNA, which means we end up with a different protein. And that different protein is going to have a different function. It'll be a different antibody. All right, now let's move over to cell-mediated immunity. Remember, the job of cell-mediated immunity is to destroy your body's own cells, which are infected with some type of intracellular infection, whether it be viral or bacterial. The steps involved include, start off very much like the steps in antibody-mediated immunity. First of all, a macrophage or some other phagocyte is going to engulf and intracellularly digest the pathogen, and it's going to display the antigens on its own cell membrane. Again, antigen presentation. Those macrophages are going to interact with T cells, helper T cells, with binding sites for that specific antigen and activate them. Macrophages are going to use interleukin-1 to help that process. The helper T cells are going to release interleukin-2, which in this case is going to stimulate a different kind of T cell called the cytotoxic T cell. It's going to activate cytotoxic T cells specific to this particular antigen in this infection. Those cytotoxic T cells, once they're stimulated, start to search the body for any cells that are displaying those antigens. And once it finds them, it's going to kill them. Your body cells, your somatic cells, have a different kind of MHC protein called an MHC1 complex. And on these proteins, the cells present antigens from within inside themselves. If they're not infected, they're presenting normal proteins. But if they're infected, they're going to present proteins from the virus or bacteria or whatever organism is causing the infection. This alerts the cytotoxic T cells that there's something going on inside, that they're infected. And essentially, the cell is telling the cytotoxic T cell, hey, I'm sick. You need to kill me before I spread this to other parts of the cell. So the cytotoxic T cell is going to use a receptor called a CD8 receptor to attach to the MHC1 complex on the infected cell. Once it's attached, the cytotoxic T cell is going to release proteins called perforins, which are essentially going to poke holes in the infected cell and help to kill it. Once the infected cells are destroyed, you don't really need all these T cells floating around. So again, they undergo programmed cell death or apoptosis. But just like what happened in humoral immunity, this process generates some memory cells, which will be with you probably for the rest of your life. And they essentially make you immune to this particular pathogen or antigen if it gets into your body in the future. Here we can see a somatic cell displaying or presenting foreign antigens on its MHC1 complex that binds with an activated cytotoxic T cell at the CD8 receptor site. Once that happens, the cytotoxic T cell is going to release those perforins, Those are going to perforate the infected cell and kill it. Once the infection's over, the cytotoxic T cells go through apoptosis and die, but you're still left with some memory T cells, which will protect you from future invasions of this particular pathogen or antigen. So again, let's look at this diagram. If you understand this, you can pretty much explain your specific immune response. Let's start out on this side, the humoral or antibody-mediated side of the response. You're exposed to an antigen. That antigen is engulfed by a macrophage, which is going to break it down, and it's going to present it to a helper T cell. That helper T cell is now active. It's going to release interleukins, and it's going to stimulate B cells or B lymphocytes, but only B lymphocytes specific to this antigen. Once they're stimulated, those B cells are going to go through clonal expansion. Most of the B cells are going to differentiate and become plasma cells, which will make antibodies specific to this antigen. They attach to it and help your body to rid itself of that antigen. And remember that once the infection's over, these plasma cells are going to go through apoptosis and die away. But also remember that some of the activated B lymphocytes didn't become plasma cells. They became memory cells, which essentially will make you immune to this antigen in the future. Also remember that this process can be stimulated by free antigens binding to receptors on the B cells. Now let's look at the other side, the cell-mediated side. So an antigen, again, is in your body. It's engulfed by a macrophage, which interacts with the helper T and stimulates it. That helper T is specific to this antigen. It's going to release interleukins, which are going to stimulate cytotoxic T cells to go out and look at all your cells and look at the MHC1 complexes on your body cells to see if they're displaying this particular antigen. If they are, those activated cytotoxic T cells, which have cloned themselves and now are active down here, they're going to secrete perforins and kill your body's infected cells so they don't spread the infection. Once the infection's over, they go through apoptosis. Most of them do and die away. But remember that some of the activated T cells never became cytotoxic T cells, at least the active ones. They became memory cells, which again will make you immune to subsequent invasions of this specific antigen. Again, if you can explain this diagram, you have a pretty good understanding of the specific acquired or adaptive part of your immune response. We've already sort of alluded to what we're going to talk about here, but we're going to talk about the primary versus the secondary immune response. The primary immune response happens the first time you're exposed to a particular pathogen or antigen. This response is fairly slow, and the number of antibodies that are made is fairly low. You're going to be sick for a few days the first time you're exposed to a new germ. This process does, however, create memory cells, which will be with you in the future. So let's say that years later you're exposed to the same germ, the same pathogen or antigen. Your immune response, the second, the third, the fourth, the fifth time, is called the secondary immune response. And because of the presence of those memory cells, this response happens much faster, and the antibody concentrations that are made are much, much higher. This response is typically so large and so fast that you never experience any kind of symptoms from infections after the first one. So on this next slide, we'll see a diagram that sort of shows this. So notice the primary immune response takes it a little while to get started. The antibody levels never get too high. Probably by this point in time you're getting well. Those plasma cells starts dying off. Your antibody levels drop. Here you're exposed again. This could be years later. Because of the memory cells, the number of antibodies, the concentration of antibodies rises very, very fast, and it rises to a much higher level. This secondary immune response happens so much faster and is so much larger than the primary response that you typically aren't going to get sick the second, the third, the fourth time you're exposed to the same germ. And that's the immune response, and I hope that helps.